Thank you, Eric. I think based on tradition, we'll ask you to read resumes and credentials online, and we'll just dive into our conversation. So, this is the best panel. It's the end of the conference, so we are the dessert for everything you heard today. And this panel is about impact and how we bring products from bench all the way to the marketplace to make humanity a better place. So with that, we'll start with Anne. And Anne, you are in the middle of fundraising for a new company, a new UCLA spin-out. So tell us a little bit about this company.
Yes, thanks, Amir. So my name is Anne Andrews. I'm a professor here at UCLA. And I have a startup company called FemSync, where we're developing the first ever 30-day patch that will be able to measure women's most important reproductive hormones continuously and non-invasively in sweat. We know, of course, that women make up more than half the folks on the planet. They're cycling for half of their lives, and yet they have very little information on their actual hormone levels, which we can measure directly and continuously. I was just in the women's health panel upstairs, and so I'll just illustrate a quick example of many of how this will be useful to improve women's lives. We know that women are delaying their first pregnancies as they are developing their careers, taking care of aging parents. And this runs up against the time in which their fertility is declining. This is creating a huge need. Being able to know your hormone levels, not just at the time you're trying to get pregnant, and by the way, we can tell you exactly when you're ovulating and whether you have ovulated, but knowing how your hormone levels are changing as an N of 1 individual over time will allow you to make decisions about your fertility. For instance, monitoring in your 20s and knowing how your estrogen levels are changing will give you information at the various early stages of declining fertility. Women tell us that if they had this information, they would make different choices in their 20s and 30s. They'll know when they're going into perimenopause, another huge swath of women that are truly underserved. We're really excited about the fact that this technology was developed in my laboratory. I'm a neuroscientist, so the sensors that we developed were actually first developed to measure brain neurochemicals, so they're really excellent figures of merit that no other sensors have. And with the support of UCLA TDG, we've spun out FemSync, and I'm really excited to tell anybody here more about it.
Great, thank you. So Dino, we heard many qualified entrepreneurs today, but I think you're, as far as I can tell, you're the only one who is a faculty member at UCLA, but you're also a serial parallel entrepreneur. What's the DNA in your lab that allows you to do that?
Yeah, so thank you. So I'm a professor of bioengineering, and I think one of the key things that's important as we try to tackle many different problems is interacting and interfacing with folks that we don't normally talk to, that have problems, and across medicine, and across, it could be in neuroscience, it could be in humanities. Talking to folks to understand their problems, I think that's one thing that that we practice day to day in the lab. So any new project that we're starting, we're first trying to reach out and understand, is this something that, as Eric was mentioning, that we can see as a product at some point, right? Even in an academic lab where others are judging by papers or grants that they receive, we're thinking a little bit further down the road, but we're realizing, papers are part of that step, but that's not the final step, right? So I think that's been critical. The other key piece has been that in order we can't do everything, right? So we need to build teams, and so one of the things that's really important for us in our lab is I really mentor the students to take the lead on companies. And now I think across 5 or 6 companies, I have a student that's either a CEO of the company, a CTO of the company, or some key figure that's bringing the technology that they developed in the lab out into the real world. Why is that important? Because at the very earliest stages, when they're in the lab, they're thinking, that could be me in the future, and so I should do robust exciting work that I am actually confident in, that I believe in the lab, and not just publish the next paper, so that they can bring it out and actually bring it on as their career develops. So I think that brings this entrepreneurial energy all the way, back from the future into the present in the lab.
Awesome. We need to replicate this model more here in UCLA. So Brad, Katmai, you're tackling a really difficult problem. Tell us about it.
We are. Amir, good to be here. Thanks, everyone. I'm president of Katmai Pharmaceuticals. It was actually founded by 3 current ongoing faculty members here at UCLA. Quite an interesting grouping in the sense that they represent clinical, chemistry, and biology. So a very comprehensive approach to what's been an intractable issue in medicine. The company really was founded or came out of frustration, and that was the lack of any meaningful clinical progress in one of the last frontiers of cancer medicine, and that's cancers of the CNS, or brain cancers that actually originate in the brain. I think it's been 22 years, 23 years since the last approved drug that remains standard of care. So the company really from day one has focused on, and I will say hyper-focused, on being best in class at solving 2 fundamental issues. One is, how do you get drugs at therapeutic concentrations across the blood-brain barrier into the brain, and how do you also selectively target oncogenic or genetic drivers of brain cancers? I'm pleased to say that we're actually cleared phase 1. We actually have 2 trials underway, one here at UCLA, a second one at Sloan Kettering, both in 1B. And interestingly, because of the challenge, as you say, Amir, of funding and traction, wholly funded. In fact, we've secured from day one $65 million, $70 million of non-dilutive funding. It's all been from philanthropic, licensing, and grant sources. And that's really our current focus and domain of expertise.
So Brad, the traditional model for spin-out companies would say, you're in the clinic, you basically sever ties with your scientific founders and academic institution because you're on your own. How does that look from your perspective these days?
Yeah. It's a great question because I think we took both a pragmatic and a gutsy approach to it. Very simply, we decided that we would make everything from drug discovery to translational work to clinical study work centered here at the university. So our company is virtually virtual, and we have remained this sort of immense and intense partner of UCLA. What's been phenomenal about it, and I think I spend as much time talking to TDG as I do talking to our founder group, is the kind of the force multiplier, the amplification that we get. So we not only tapped into the university resources, but you have to remember academic talents also have huge global collaborator networks. And very specifically, that's how we got picked up by Sloan Kettering, who happened to be a very close collaborator, some of our faculty here at the university. So with that, we've been able to sort of build these building blocks in order to advance molecules to and through the clinic. And I'll say the mantra these days is lab to clinic, and accelerating that or compressing that time frame while upping the probability, translational success. And the key for us has been actually doing it here to and through university resources.
So, Dino, you're in the School of Engineering, yet one of your companies has a product in the market
Yeah.
solving a critical diagnostic problem for the medical profession.
Yeah.
Can you elaborate on that project?
Yeah. So the first company that we founded that came out of technology here at UCLA was a company called Cytovale. It's tackling sepsis. And actually, I'm sure many of you have either your family members or yourself, actually, of us 4 here, 3 out of 4, it was a family member or themselves were affected by sepsis. One of the main killers in the hospital system. This is when your immune system aberrantly attacks your own body after an infection. And every hour, 7% increase in mortality. And there's no diagnostic that measures how your immune system is, what state it's in, that tells you have sepsis, until we developed this test with Cytovale. And this actually started as an accident here in the engineering school, where we were starting to collaborate with folks in medicine, in cytopathology actually, looking at cells that are in the fluid around the lungs and pleural effusions. And we found that these white blood cells in that fluid that during inflammation states was squishier. They deformed more when we applied pressure to them using microfluidics. And so that engineering, this kind of accidental discovery, it wasn't very helpful in the application of looking at pleural effusions, but we thought, okay, if we can measure the state of these white blood cells, could we apply it to these emergent conditions like sepsis, and where every minute counts. And so this test, because it's not a molecular test, we're not measuring any molecules, DNA, proteins, et cetera, we're just squishing cells, can be done in less than 10 minutes. So really rapid, from a tube of blood, you get an answer, this patient has sepsis or not. It's now, it was FDA cleared a few years ago. It's in hospitals starting in Louisiana and Alabama, Texas, Florida, Minnesota, not here at UCLA, UCLA Health, yeah, they're maybe soon. But over 65,000 patients have been tested so far. In the hospitals that have adopted it, 40% decrease in sepsis-related mortality on average, and reduction in the cost per patient, reductions in lengths of stay, and less blood draws, less for blood cultures. So lots of benefits to the hospital system, to the patients, to the doctors. So it's really exciting. I went to visit the first hospital. It started here at UCLA. Actually, the first tests we were doing here with the emergency department. But the first hospital that adopted it was in Louisiana. I went to visit them, talked to the doctors there, and just heard the stories of patients that they thought were septic or not, and that they were not. And then they went through this diagnostic journey and they found the real cause and how they could save these patients. That was worth the kind of 10 years of effort, and it's been an amazing journey. Really proud to be part of the UCLA family with that technology.
So and you also had a very interesting journey with some false starts along the way. So talk to us about the power of perseverance in that journey.
Absolutely. I loved what you said, Dino, and about thinking about hard problems and trying to move the needle, right? And that's what we do in my laboratory. And the first, sensors we tried to spin out when we discovered that these brain sensors could be used on the body and could be widely used was around cortisol for stress. And so we had 2 early attempts to try to commercialize around that. And we're still doing that, but what I learned really was that it's not just having an A-level technology or an A-level science, but it's about having an A-level team and people to support you along that way. And so, persevering through trying to put teams together around building a company and then finding out that maybe they weren't they weren't a good match, and then moving on quickly is definitely a lesson I've learned as a first-time entrepreneur. And again, having the support of TGG, they worked with me for 2 years on pitch coaching. They beat a lot of silliness out of me to the point where we won pitch competitions, but that really helped us define our market, think about, who we were gonna market this product to, turn something that, clearly had wide applicability, but make it something that was attractive to investors,
So all these technologies are not born overnight, right? So talk about the connection between the application and basic science.
Yeah. I mean, for us, the development of the sensors is backed by over 25 years of basic research. We were really trying to solve a hard problem, which was how we could use transistors and electronic signal transduction in wet, salty body environments to sense, in our case, neurotransmitters. So we were really always motivated by the biology. That work was backed by $10 million in federal funding. And so, it was really a long trajectory to get where we are today. And that, having support for that, again, was crucial.
Dino?
Yeah. So our sepsis test started with grants from the National Science Foundation, from DARPA, Defense Advanced Research Projects Agency, from BARDA. Some of the original work was just to develop this technique we called deformability cytometry, which is, what if we squeeze cells and measure them? Could we get any information out of that? Very basic, right? No one, it seems, the company's not going to, actually, we talked to companies, no one wanted to fund us. So, especially in this time of kind of uncertainty in research funding, these long trajectory investments that the government makes are so critical, because without that, we wouldn't have this IntelliSub test now.
And Brett, you still have an active research project here in UCLA.
Active
Research project in UCLA.
Oh, one of the, just picking up on what Ann and Dino said, one of the things that really fascinates me, and I love about my whole career, I've worked kind of alongside academics and taking pretty bold, leaps in the lab, a lot of them not working, most of them not, and fortunately the ones that do. The team we have, here we were driving and are driving a molecule through the clinic, and I don't know, 6 months ago, 8 months ago, I'm having a conversation with one of our co-founders, and he said, we're working on a super molecule here. And I go, really? What, I called about the one we're already in development. He goes, that's doing great. We got it in the clinic. We've got the pros behind it. But I've got an idea. There's kind of a holy grail in targeting oncogenes. And the problem is on-target toxicity, meaning you might knock out the bad guy, but there are a lot of bystander good guys that get hurt. And so that toxicity, skin, intestinal, whatever. And he said, we're working on a new class of molecules that are absolutely selected for the mutated form of the oncogene, but are really not having an effect on the others. All that's here at UCLA. All of it, frankly, was not even planned in my playbook, but it was really on the initiative of faculty here who just kind of never stopped thinking, doing, and solving stuff. So call me a pretty big fan. I think there's so much more alliance work that can be done between industry and academia that just is not yet really being taken to where it should and could go.
Okay, Dino and Ann, we're 90 seconds before dinner, and we won't stop dinner. So, quick answer, lessons learned from your journey.
Bring as many people along with you as you can. Yeah. Was it only like one sentence, or was I? Oh, I have more time. So I think also it's always the unknown unknowns that get you. Maybe that's another one, right? So it's the things that you thought you knew that you never really did know. So and be humble. Yeah. So I was. My field is this microfluidics field. That's where I started. And every 5 years, I kind of change fields. But this microfluidics field, I was like, oh, we know how to make microfluidic devices, and we are trying to manufacture this for the sepsis test. And the first manufacturer it all blew up, and it was. We had to go and find others, and it was a big waste of time. So what I thought I knew, I didn't know.
And Dino, you chose not to be active CEO. You chose to stay here.
That's right, yeah.
Why is that?
Yeah, I think that I love being here and being able to come up with the next ideas and explore all those paths and then hand it off when it's time to the right person to bring it all the way to the finish line, yeah.
And from your journey?
I would say one of the biggest lessons is that when people start telling you that you can't do something, that's the time to maybe think about doubling down. You know you're on to something. So.
Great. Eric, I think we'll be the first panel to finish on time. Thank you very much, and we'll be here for dinner.